Hi, everyone, and thank you for joining today's webinar. My name is Aaron Pomerantz, and I'm the Associate Director of Global Segment Marketing here at Oxford Nanopore Technologies. Before we hand things over to Anthony and Matt from Plasmidsaurus, who will take us through the technical details of plasmid and AAV sequencing and analysis, I'd like to spend a few minutes sharing updates on how Oxford Nanopore is supporting the broader biopharma and biotech community and walk through some of the recent performance improvements across our sequencing platform, helping teams like yours get richer, faster, and more complete insights across a range of applications. So let's get started. At Oxford Nanopore Technologies, we're ushering in a new generation of single molecule sensing technology, one that's designed to enable the analysis of anything by anyone, anywhere. Our multi-omics analysis platform already supports integrated genomic and epigenomics within a single assay, as well as transcriptomics for bulk cDNA, single cell, and direct RNA sequencing, with proteomics and metabolomics in development. This validated technology is implemented globally with over 14,000 peer-reviewed customer publications to date and growing. And whether it's in the lab or in the field, our platform delivers accessible, real-time biological insights at any scale. So just for a bit of background, let's walk through what happens during the process of sequencing with Oxford Nanopore. First, we take a strand of DNA or RNA, and a motor protein feeds it through a tiny protein nanopore embedded within a membrane. As the molecule passes through the nanopore, we apply an electric current across the membrane, which creates an ionic flow through the pore. So as nucleotides then pass through the pore, that current is disrupted and produces a distinct signal. We then detect these changes in the current in real time, and our software translates those raw signals into nucleotide sequences. Importantly, this approach means we're not using optics or amplification methods. We're directly sensing native molecules, including base modifications. So now we've seen a little bit how nanopore sequencing works, but how does that translate into real-world benefits? Well, there are a few. First, by providing richer insights. Because we're sequencing native molecules without amplification or chemical conversions, we can detect fragments of any read length, from short to ultra long, which enables us to capture all elements of a genome, from single nucleotide variants to large structural variants, and even epigenetic information within that assay. Second, enabling faster time to result. Our platform can stream data in real time, and that means you don't have to wait for a full run to complete. You can start actually analyzing results as the data is being generated, which can be especially valuable in rapid decision-making environments. And third, accessibility and affordability. The same core technology scales from pocket-sized devices like the MinION to high-throughput systems like the Promethean, which many production-scale sequencing labs are running today. And that flexibility allows teams to bring sequencing closer to the sample and to meet their volume needs. And so when it comes to innovation, we know that speed matters. In synthetic biology and biopharmaceutical development, we're all familiar with the design, build, test, learn cycle, an iterative loop where every delay in testing slows down learning, optimization, and ultimately time to market. So whether you're engineering plasmids or optimizing viral vectors or scaling production processes, we need fast, reliable data, which is essential to keep that cycle moving. And that's really where this technology stands apart. So unlike traditional sequencing platforms that require fixed runtimes or high sample batching or extended protocols, our platform offers real-time access to that information. So what we see here, as soon as DNA starts to enter a nanopore, you start generating that signal data. And then our integrated software, Minnow, can perform base calling as the experiment runs. So this converts that raw signal into FASTQ files continuously. There's also no need to wait for the full run to finish. So if you have enough data for your experiment, you can actually stop that run and you could move straight into analysis. And if you have capacity left in that flow cell, you can actually wash and reuse it for additional experiments at a later time. So on the right, we see an example of how that real-time data generation can translate into applications such as real-time detection of microbes within a sample as that data accumulates. And in the context of biopharma development, we see users like Dr. Jianing Liu at Vertex on how implementing Oxford Nanopore for clone validation reduced their turnaround time from 2 weeks to just 1 day. So this real-time capability enables innovative service providers like Plasmidsaurus to achieve such rapid turnaround time from receipt of their samples to generating reports for their users. And over the last decade, we've been delivering and enhancing new products, not just in hardware, but in chemistry, software, and informatics designed to improve the overall performance of the platform. We've now advanced to greater than Q20 single molecule raw read accuracy, delivering exciting improvements through chemistry and base calling updates while using the same core devices. And because the platform is unified by the same nanopore chemistry, we can support a wide use cases and throughput needs. So on the bottom here, you'll see some of our suite of devices, including personal sequencers like the handheld Minion, to desktop Promethean 2 Solo, to scale-up options like the Gridiron and the Promethean 2 Integrated, all the way up to high output systems like the Promethean 24, capable of large-scale, high-throughput projects. So now let's take a closer look at some of those developments in single molecule raw read accuracy. Here we see performance from our base calling algorithms on individual reads for DNA and RNA. On the left, we're looking at native DNA raw read accuracy. So using our latest chemistry, which is KIT V14, and here we use two different base calling models called the HAC and the SOUP models. Our latest DNA models are now achieving modal Q scores above Q26, or well above 99% single molecule accuracy, which is fantastic. So that's a major improvement over previous generations, and it just continues our upward trajectory in raw read base calling. Oxford Nanopore is also the only technology on the market today that can directly sequence RNA molecules, so we're not converting it to cDNA, we can read the RNA directly. And on the right, we see that raw read accuracy for native RNA sequencing is achieving Q19, approaching that 99% single molecule accuracy. And I think one thing that's important to note here too is that these improvements are in large part due to software-driven improvements, meaning that existing users can benefit from higher accuracy simply by updating their base calling models without changing of their existing kits or instruments. So now that we've covered raw read accuracy, which reflects the accuracy of individual reads, let's turn our attention to consensus level, which is the accuracy of the final assembled sequence that can include error correction and polishing tools. So on the left, we see a microbial genome that was assembled by short read sequencing. So this winds up highly fragmented. Here we have 110 contigs, and this was unable to completely resolve the bacterial genome from the extrachromosomal plasmid. And this is a common challenge for short read sequencing technologies, especially with complex or GC-rich genomes. In the center, however, we see the result of an Oxford Nanopore only assembly. So just two contigs, one for the closed bacterial genome and one for that plasmid. And that's the power of long, accurate reads. So this enables complete genomes and it provides the full picture. On the right, we see a case study from Ryan Wick at the University of Melbourne, showcasing a bacterial genome assembled using Oxford Nanopore reads and recent base callers. The final assembly achieved Q63 consensus accuracy, or 99.99995% identity. This level of accuracy makes Oxford Nanopore ideal for applications like plasmid and viral vector characterization, where you need complete, confident assemblies from a single sequencing assay. And one of the other powerful aspects of Oxford Nanopore's platform is that we can go beyond just the canonical bases and detect base modifications directly in native DNA or RNA. That means you're not just seeing sequences of A's, C's, G's, and C's, you're also capturing biologically meaningful modifications like 5-methylcytosine, 6-methyladenine, and 5-hydroxymethylcytosine, all in a single experiment without a need for any extra sample prep steps or chemical conversions. And this chart here shows progress that we've made in that base modification calling accuracy over time across different contexts and modifications, both in prokaryotic and eukaryotic samples. This level of modification detection is really best in class and opens up new opportunities in epigenetic research, as well as applications such as messenger RNA vaccine QC, where those mods are quite important. Now that we've covered technology and performance updates, let's pose the question, what if a single platform could speed up your discovery process and reduce failure risk, from R&D to manufacturing? Oxford Nanopore provides solutions across the biopharma value chain, starting with biomarker target discovery. So here, our multi-omics capabilities can allow researchers to combine genomic, epigenetic, and isoform-level transcriptomic insights, and this can really help to accelerate discovery of novel biomarkers and targets. Now, as we move into lead identification and construct validation, our platform supports verification of materials such as engineered plasmids, synthetic DNA, and RNA constructs, ensuring design accuracy and preventing costly errors downstream. In vector and cell line characterization, we enable full-length genome analysis of viral vectors like AAVs or lentivirus, which we'll hear more about today, and supporting engineered cell line validation, helping confirm stability and transgene integrity. And then finally, in biomanufacturing QC, our platform can really help to profile a number of critical quality attributes, including comprehensive sequencing identity and integrity to reduce regulatory risk and accelerate paths to approval. Now let's zoom in a little bit more on gene therapy applications, which will be the focus of this webinar. Here we provide powerful insight across key stages of gene therapy development to help teams make confident decisions and keep their programs on track. With whole plasmid validation, it's important to ensure that what you designed is exactly what you produced. So the ability to generate long reads enables you to validate full plasmid constructs in a single read, which can reveal structural variants, unwanted mutations, or contaminating sequences that could compromise downstream vector production. So next, with viral vector integrity, this is a critical checkpoint for AAV workflows. And within our platform, you can sequence full-length genomes and assess vector quality in detail. This allows you to detect things like truncations or rearrangements or sequence impurities that could affect your vector potency or consistency. And lastly, after that payload is delivered, thorough gene editing characterization should take place. Here, long reads again allow you to determine whether that edit was successfully delivered to the intended target site, and perhaps just as importantly, whether any off-target effects have occurred. So the need for comprehensive sequencing isn't just theoretical. When it comes to high-value biological materials, what you're missing can truly matter. Hidden errors in samples can jeopardize entire projects, they can compromise reproducibility, and delay time to market, often without users even knowing they're there until it's too late. So let's take an example. A recent study with Bai et al. Analyzed thousands of lab-made plasmids and found that nearly half contained undetected sequence or design errors. And to me, studies like this should really be a wake-up call for anyone in this field, as these are the very reagents used to express your genes of interest and produce your viral vectors, and ultimately advance your pipeline. In the antibody space as well, others have pointed out that most antibodies used in research are actually never sequenced. A researcher named Kathleen Zeglinski and her colleagues did a recent study on this. And as they put it, sequence-based validation of antibody reagents can be a valuable way to check what you're using is actually what you think it is. And this, again, is where comprehensive profiling with Oxford Nanopore can truly make a difference for your group, giving you complete visibility into the constructs, the vectors, and the biological reagents that you depend on so that you can spot problems early before they cost you time and resource. So why is it that so many critical errors in these materials have gone undetected for so long? Well, part of the answer could lie in the limitations of legacy technologies, such as Sanger sequencing, which, despite still being commonly used, often can't capture the full biological complexity that today's applications demand. Here we see a few real-world examples of how Sanger sequencing can miss critical biological information due to inherent limitations of the technology. So in the top left, we can see that multiple peaks are coming about in the same locations. And this is a sign that more than one sequence is likely present in that reaction. That could perhaps be a colony contamination or a mixed plasmid population, both of which Sanger can't resolve cleanly, and the sequence reaction has therefore failed. In the top right, we see the sequence abruptly terminates, and this can be due to secondary structures like hairpins or high GC content regions, for example, the inverted terminal repeat or ITRs of AAV vectors, which traditional Sanger runs often fail to read through. In the bottom left, the sequence becomes unreadable after a stretch of mononucleotides, and this is likely due to polymerase slippage, a known failure point for Sanger, especially in homopolymer-rich regions like poly A tails in mRNA constructs, for example. And finally, in the bottom right, we see a sequence that starts off clean, but then becomes mixed and ambiguous downstream. And this can happen in a polymorphic region or perhaps after a random mutation was introduced during the cloning process. And again, Sanger typically fails to resolve this biological complexity within a sample. So in all these cases, you're really not getting the full story, and that is a risk when you're working with gene therapy vectors or synthetic constructs or these critical QC checkpoints. So after seeing the problems that can arise from undetected errors, let's talk a little bit about what you can actually do about it and why it could be time to rethink traditional sequencing workflows. Here we see a comparison of legacy Sanger sequencing approaches with Oxford Nanopore's approach. So with Sanger, you're only getting a partial sequence coverage, and that often means you're relying on primers, or if you want to capture the whole construct, you have to do a process of manual primer walking. And both introduce cost and time delays, which has the potential to miss important regions of that construct. In contrast, nanopore sequencing delivers full plasmid reads, allowing you to verify that all components, including the insert and the backbone, are free of mutations. You don't need any primers, and there's no guesswork. Even repetitive regions, structural variants, dimers, deletions, these can be resolved with this long read-based approach, which may otherwise have gone missed by short read-based methods. It also works across a wide range of plasmid sizes, construct complexities, including GC-rich, and can identify mixed populations or even unexpected contaminants, something that Sanger would completely miss. And we often hear from our users that other contaminating plasmids often popped in there at some point, maybe from a vendor or maybe from the lab process itself. So as we've already seen, Nanopore data is also fast. This makes it really suitable for these routine quality checkpoints, as well as construct validation during rapid development cycles. When it comes to amplicon sequencing, I'd also like to make the case that you no longer need to be limited by constraints of legacy short read approaches. So on the left, we see that traditional Sanger sequencing offers limited short amplicons. So typically these are under 1,000 base pairs, and these require primers for each read. With amplicons, it can also struggle again within those repetitive, GC-rich, or structurally complex regions, and often leaves critical gaps or ambiguities. And if you're working with multiple targets, these costs and timelines can start to increase quickly. Now, if we compare that with the advantages of an Oxford Nanopore-based approach, there's no upper limit on the amplicon length. So whether you're working with a gene target that's several hundred or even several thousand base pairs long, you can sequence that amplicon stretch if you can generate that amplicon. You can resolve entire targets in a single read. So this again can include those complex regions. And then with barcoding based approaches, you can multiplex high numbers of amplicons in a single run, making it overall a faster, more scalable, and more cost-effective approach for amplicon screening. Last but not least, let's talk a little bit about why the technology is uniquely suited for characterizing viral vectors such as AAV genomes. So short read technologies have long struggled with this task, and on the left you can see why. Traditional methods generate reads of just a few hundred base pairs long, and this means they can't span the full AAV genome. And they often fail to read through the inverted terminal repeats due to their high GC content and complex secondary structure. This makes it difficult to distinguish between full-length genomes and partial truncated versions, let alone capture the full true diversity of the sample. On the other hand, an Oxford Nanopore-based approach produces long single-molecule reads that span the entire AAV genome from ITR to ITR. That means that you have the ability to gain insight into several of those critical attributes all at once, including sequencing through those GC-rich ITRs, capturing complete genomes, the ability to detect residual plasmid or host DNA contamination, as well as resolve vector heterogeneity in your sample with that single molecule resolution. So to wrap up, collectively, I hope you've seen that this is what makes Oxford Nanopore a differentiated technology. We aim to provide a unified platform that enables this ultra-rich and timely biological information. And with innovators and partners like Plasmidsaurus making this technology even more accessible and tailored for real-world biotech applications, it's never been easier to get started using this platform. So with that, I'll hand it over now to Anthony, who's going to walk us through how Plasmidsaurus is applying Oxford Nanopore sequencing to accelerate gene therapy. Over to you, Anthony.
Thanks for having us. I'm Anthony Berendt, and presenting with me today is Matt Working Team. We're a scientist and a software engineer and informatician at Plasmidsaurus. And we're very happy to show you some of the services that Plasmidsaurus has been providing for a long time, as well as some newer ones that lets you see what's really in your gene therapy product. So other than being dinosaur themed, Plasmidsaurus really wants to sequence everything. And we've been continually expanding our services to include new product classes, new types of DNA, and new complexities of DNA. Plasmidsaurus originated from our co-founder and CEO, Mark Buddy, being very frustrated by what Sanger sequencing could do for him. And this was especially problematic because a project he was working on involved making hundreds of plasmids that needed to be sequenced by Sanger sequencing. Eventually, he tried using Oxford Nanopore technology and realized not only would this accelerate his own work, but that he could start helping other scientists. So Plasmidsaurus really was born in a garage, and Mark started processing and sequencing samples for other people. This was a service founded by scientists for scientists. And one of the things we keep on trying to do at Plasmidsaurus is providing results that we would be happy with if it were our own experiments and own projects on the line. And here's how it's going. We've expanded to 10 different sequencing labs. We just opened one in Singapore. We have over 800 drop boxes. In fact, this slide is probably old. We have over 1,000 at this point, serving 23 countries, and we're seeing continual growth every single month. We want to sequence everything, and we want to do it fast. We accelerate your research by providing high accuracy results with innovative approaches that no other provider does. This includes bioinformatics pipelines and visualization tools that get you your results and let you see what's really happening in your sample without having to do your own analysis. And we combine this with a global logistics network that get your samples to our sequencing hubs in just hours, and then those samples sequenced just hours later. So recently, we took a set of plasmids, some that challenged us, some that are pretty easy to sequence, but all being very representative of what customers regularly send us. And we sent them out to other full plasmid sequencing providers. Some of these, such as vendor A and E, I'll let you guess who they are, but they are globally distributed. And not only could we sequence over 90% of these plasmids, including some very challenging, highly repetitive ones, and do this better than any of our other competitors. But we could do this with a turnaround time of 4 to 6 hours upon receipt of these plasmids, whereas the other sequencing providers took hours, even the better part of a day, to return these results. So not only can we sequence plasmids more accurately and more quickly, but we can return these results faster than other services. And with specific relation to gene therapy and AAV production, we actually sort of forget that we can sequence through high complexity sequence such as AAV ITRs without problem. Because of the way Oxford Nanopore chemistry works with the strongly displacing helicase, we can read through these regions just like they were a protein coding sequence. So some services will charge up to $200 for special Sanger sequencing through these regions. This is part of our standard package for $15 and next day results. Not only that, but our sequencing can resolve very complex rearrangements of plasmids. So the data here on the right is an example of a plasmid that is recombining with itself and creating a dimer at a low frequency in the population. These results are extremely useful and important when you're thinking about scaling up your plasmids, where small amounts of contaminants can later become big problems in downstream work. So we really do try to sequence everything. Even though we started out with plasmids, and that's still by far our most popular service, we've expanded to sequencing PCR amplicons. For the cloners out there, we can allow you to skip the mini prep step and send us colonies directly where we can extract the plasmid and sequence it as if it was a purified sample. We also offer bacterial genome and eukaryotic genome sequencing, as well as a highly adaptable custom service. So if you have a sample that is amenable to Oxford Nanopore chemistry, we can accommodate it and sequence it. So with specific respect to gene therapy, gene therapy is a race. Not only are we racing to get treatments out to patients, many of these patients having few or no therapies available at this time, but you're also working against funding deadlines, you're working against investor expectations, and as a distant third or fourth, potential other competitors. So With AAV production, because it takes a lot of money, time, and energy to even make small AAV samples, people tend to produce them, put them into in vitro experiments, screen out potential candidates, scale this up so that they can do in vivo experiments. And then once they've down selected, that's when they tend to do scale up production. And this is the first time that AAV is sequenced. And this is understandable. The services that have existed up until essentially we've offered a new chemistry and approach take weeks to do, involve very large samples of virus to the point that companies are making a choice of whether they do a few more in vitro experiments or they send one batch out for sequencing and then have to sit around for weeks to months before they get these results. All this time, there is runway burn down, there's full-time equivalent salaries to be paid, and slow data is just quite frankly very expensive. So when AAVs are finally sequenced, usually at the point where they've been scaled up, often part of the establishment of chemistry, manufacturing, and control departments, often there's some panic. At this point, people realize there may be contaminants or subgenomic variants in viruses that they have already carried through a pretty intensive research program. This, again, generally causes people to pause and have to reevaluate not only their transgene design, but even their production methods. So how do you ensure quality by design in AAV? How do you make a proactive process that guarantees quality rather than doing it at the very end of the process? And Plasmidsaurus offers a solution here, which is a fast turnaround, affordable AAV sequencing that can be done at every single step because we require very little input. So just so that we're all on the same page, and also for the people who are not gene therapy experts that may be watching this presentation, adeno-associated viruses, or AAVs, are single-stranded DNA viruses. This DNA is packaged into a non-enveloped icosahedral capsid. They're ideal for human therapies and in vivo experiments due to their low immune response. Usually their genomes are non-integrating, as in they exist as episomal arrays, but they do integrate at a low frequency. And these viruses can target dividing and non-dividing cells. So things like neurons and muscles become accessible to gene therapy. And conventional AAV production has been around for about 30 years at this point. This involves what's called triple transfection of a payload plasmid. So this is your intended transgene flanked by inverted terminal repeats, or ITRs. The rep and cap genes from AAVs in a second packaging plasmid, as well as a helper plasmid containing some adenoviral genes that are required to recapitulate adeno-associated virus life cycle. These are delivered by transient transfection, after which the cells are lysed and harvested. There's a clarification and purification step, and then usually final formulation. Because of the way we've designed our AAV sequencing method at Plasmidsaurus, we allow for sampling at each one of these production steps and verification of the quality of your material. So, although we can't really help out with the very earliest stages of the research process, such as hypothesis generation or designing your transgenes, there is a Oxford Nanopore-based solution for every other step in the R&D process. And this is really important because AAV production is error-prone. The initial plasmid starting materials, such as the payload plasmid, can have instability. So it's been well described that ITRs can accumulate deletions, and this changes AAV packaging. These ITRs, because they're high complexity sequence, can also recombine with themselves and delete parts of the plasmid. Even the rep cap plasmids, because many of them have been modified to increase AAV productivity, have intentional duplications or deletions that can also lead to plasmid instability as gram quantities of plasmid are grown for these massive transfection projects. AAV production and purification itself is problematic. Partial payload packaging is observed, so only part of the replicated transgenes are put inside the capsid. Often subgenomic variants are seen, so the actual polymerases within the cells that are copying the payload can make mistakes. Often we see this as snapback genomes, so the polymerase will switch strands and create a short hairpin sequence that not only replicates more efficiently, but also packages efficiently. And these contaminants can start taking over your AAV population. One also regularly sees what are called reverse or inverse packaged plasmid backbones. So the ITRs in the intended payload backbone actually package the bacterial resistance and origin of replication gene. So let me talk specifically about AAV genome sequencing by Oxford Nanopore at Plasmidsaurus. Plasmidsaurus has developed a unique ITR-focused library construction. And we did this because conventional long-read AAV sequencing library prep is dependent on something called end repair. So end repair involves polymerase fill-in, nuclease chew-back, and an A-tailing enzyme, as well as kinase, to generate double-stranded ends of DNA that have an A overhang and a 5' phosphorylated nucleotide. And these are required for ligating conventional sequencing adapters onto these DNA. Because of this combined activity, you tend to see a lot of short-read artifacts generated that are not representative of the integrity of the AAV library as determined by other methods, like denaturing gel electrophoresis. So at Plasmidsaurus, we developed a new way of generating AAV libraries with the aim of sequencing native AAVs. And the way we do this is by direct ligation of our sequencing adapters to ITR-containing sequences. This works on both single and double-stranded DNA, and most importantly, no second-strand synthesis, annealing, end repair, or fragmentation is involved. So what gets sequenced is native AAV DNA. One read is one native AAV genome. And we combine this with de novo consensus assemblies based upon peaks of read lengths. So we look through the output of the Oxford Nanopore sequencing and try and take advantage of the long read nature of this sequencing, take reads of about the same length, compile them together into a consensus sequence, so essentially stack them on top of one another, and generate de novo genomes. This is distinctly different than most other services that just do match to reference. And we provide this in an interactive online informatics reporting portal. So what's the practical implication for AAV researchers? One, we require low input. We only need 1 × 10 to the 11th vector genomes per sample. This is 10 times less than PacBio. We can take in a broad sample type input. We've already sequenced very crude PEG precipitated AAV. These are samples that are viscous, chunky, and in one case, because it was a GFP encoding AAV, bright yellow. These types of samples are actually very challenging to other types of library prep and sequencing platforms. We can return these results in 3 business days after receipt, rather than the 2 to 8 weeks with other services. And we offer an easy interactive analysis with all samples. There's also a deeper analysis package with downloadable reads for people who really want to dig into the data. So in creating this library that is different from existing services, different from existing Nanopore and PacBio-based platforms that use end repair, we can sequence all ITR containing sequences. This lets us return results that characterize complex and chimeric novel sequences because we are not making any assumptions about what's in the sample. And our library prep is amenable to homogeneous and variant library AAV genome populations. But because we've chosen to focus on ITR containing sequences, we don't sequence host cell DNA or residual plasmids. There are Oxford Nanopore-based chemistries that use end repair or tegmentation that can characterize these, as well as amplification-based methods that are super sensitive, super powerful, and quantitative, like digital droplet PCR. So I'm going to turn this over to Matt Workington, who is going to provide a real-world example of the data you can expect with our service.
Hey, thanks, Anthony. Hi, everybody. I would like to do a quick walkthrough here of standard results that you might see for your AAV order. This is a combination of a bioinformatics workflow, and then some of our amazing front-end engineers have developed this interactive product that allows you to gain rapid insight into what's in your sample. So this is a standard report that will be included in your order. And what we've done here is we have a demo order where you may or may not see things like this in your sample, but it gives you an idea of the types of things we can detect and some of the answers you can get with this report. So when you first look at your results, you'll see that you'll get a box essentially for each sample in your order. And you'll get a number of genomes here. But what I want to start with is actually this histogram down at the bottom. So this is a histogram of all of the reads that were sequenced in your sample. And like Anthony said, we will sequence any read that has an ITR on it. So the first thing we're going to do is identify reads that have an ITR on either end. And so these, what we will assume, are the genomes being packaged. And then with those reads, we'll identify peaks within the histogram. Presumably, these reads all represent a single genome. And so you can see here we have one for a fairly large peak that's right around 3.5 kb. And then we will take these reads and do an assembly with them to see what that genome is. And in this sample, you can see we've identified a number of other peaks of lower abundance in there as well. And we'll talk about those shortly. And then you'll also see these gray reads in the background here. And I can toggle those on and off. Those are reads that have only a single ITR. And I will talk about those, the analysis we'll do with them later. We can't assemble them. We assume that they are not potentially non-replicating, coming from truncated genomes, but we have a different analysis that we can use for those. But for the assembly, what you'll see here is we're able to identify 4 different genomes in the sample. And the top one here is our expected payload. And so we'll take the consensus sequence, we'll annotate it with public databases so you can see essentially what should be on your sequence. This is a standard vector. It has GFP on it, nothing super fancy. What we'll also do is report a relative abundance for that genome so that you can see here on the side where we estimate, based on the number of reads, the relative abundance for the genome in the sample. And then you'll notice that we've got a few other ones here that are a much lower abundance. So one of the things we wanted to do with this analysis is to make it really easy to identify things that have gone wrong, so you can quickly pinpoint any issues in your cloning or production. And so here we see we have a number of other hairpin-like structures. And so we'll detect these and show you the hairpin structure so you can quickly see what's going on. Here we have a spontaneous self-complementary genome, as well as a couple other hairpin events. These are much lower abundance, as you can see on the side here, but they are presumably real genomes in the sample. You can also notice this genome at the bottom. It's highlighted in pink. What we've identified here is a possible inverted payload or reverse packaging event based on typical vector backbone sequences, such as the origin of replication. And so you can easily see that genome in your sample and its abundance as well. So that just gives you a quick overview of the genomes in your sample. Hopefully you won't see all these other events. You'll just see one nice expected payload. However, this gives you a chance to identify where things are going wrong and what's happening. Now, additionally, you'll notice I mentioned before these reads that have only a single ITR. We can't really assemble these. There's quite a distribution of read lengths. And so we have an additional analysis that's included in your download package. So when you get your results, you'll see at the top, there's a download results button. And so you'll get all this information In your download package, you'll get a GenBank file and a FASTA file that has the sequences and the annotations. You'll get text files that have the relative abundance. The other thing we will do, and that will be included in that download package as well, is we will quantify the flip and flop orientations of the ITR sequences in each genome. And so the genome that we return to you will have sort of a standard orientation, one you would typically find in a common vector, or we'll either return that or the most abundant one if there's a particular combination that seems to be more abundant. And so there'll be a file in your download package that has that as well. And then along with that, you'll get a single ITR analysis plot, a truncation plot, which I will talk about now. I have pulled up here a static plot that you will get in your download. Again, this analysis will be performed on every sample. So if you have a lot of reads that have only single ITR, this could be indicative of something, a lot of truncation events that are happening. And often you want to know where those are. And so one of the ways you can do that is to essentially take these reads that have only a single ITR, and then we'll align them to the genome that is most likely to be the primary genome or the intended payload. And then what you see here is the count of those end positions of each alignment plotted on the y-axis. And then we do that for the forward and the reverse strand. And so what that allows you to do is identify peaks where you have a lot of reads that are ending at that same location. So for this example, we can see quite easily that we have a peak in the beta-actin promoter, which is known to be quite problematic and form a lot of hairpins. And so that is quite easy to see here. So again, in this sample, there's a fair bit of truncation going on. So this allows you to identify that quite quickly. So like I said, this is included in your download. And if you don't have an account and you sign up for a new account, you will get some demo data in your account that will have some data like this that you can play around with on your own. So yeah, there you have it. That's their standard analysis. And now I will pass things back over to Anthony.
Thanks for that, Matt. At Plasmidsaurus, we really are aiming for sequencing everything. We have solutions for the entire workflow, from building your initial transgenes to sequencing your AAV genomes, and at every step throughout that. But for the general molecular biologist, we have services that if you're interested in DNA sequence in your sample, we probably can already address it. And if we don't sequence your type of sample, or you think you have something pretty weird and interesting, get in contact with Plasmidsaurus. We will see what we can do for you and treat it as an R&D project. So if you'd like to know more, you can sign up for a new account at plasmidsaurus.com and get real-world example data in your account. So it'll actually be the data that Matt just presented, as well as example plasmid sequences. If you have questions, emailing support@plasmidsaurus.com will get your emails rapidly forwarded to our scientists. So we have experts on pretty much every sequencing platform we offer, and we try to get back to you in just a couple of days, if not faster. The link on the screen is a link to our new AAV product page with detailed FAQ written by our PhD level scientists. The QR code on the bottom left links to our AAV product page, and the QR code on the right of the screen links to Oxford Nanopore's Biopharma platform. Thanks for your time, and happy sequencing.